present about, as Christina has already said, by my title, that is to study the XMN1 gene and the beta-globin gene haplotype pattern of descent of set of linked alleles occurring on same chromosome in northern province of India. Disorders characterized by reduced or absent amount of hemoglobin, the oxygen carrying protein inside the red blood cells, and the worldwide distribution of hemoglobinopathies. So most common inherited blood disorder in the world, 250 million people are carriers of potentially pathological gene, and 3 lakhs infants are born with a major hemoglobinopathies. As I said, like it's an autosomal recessive inheritance. This figure very well explained. Like if a father is a carrier and a mother is a carrier, the chances of having thalassemia major child is 25%. So what happened with thalassemia? What happened with this globin gene production? Actually, thalassemia is of 2 types, beta thalassemia and alpha thalassemia. Alpha thalassemia gene is located on chromosome 16 and beta globin gene is located on chromosome 11. And that's how it moves forward. Like on chromosome 16, Grover 1, then Portland 1, Grover 2 in embryo. And when embryo grows to fetus, it's HbF. And when it comes to adult, it's HbA and HbA2. And when there is a mutation in either alpha or beta gene, that leads to alpha or beta thalassemia respectively. So as I mentioned, this slide shows alpha thalassemia types. Couple of them are silent carrier, and carriers of any genetic disorders are healthy, normal individuals. And then comes trait, hemoglobin H disease, hemoglobin Bart's, hemoglobin constant spray. In beta thalassemia, major concern about this study is about beta, reason being beta thalassemia is 3 types, like trait, minor, healthy, normal individual, intermediate, and major. Major is major concern, reason being they are totally dependent on blood transfusion and due to regular breakdown of RBCs. So splenomegaly and hepatomegaly is a common problem for them. And for the treatment of this, chelation therapies has been provided to the patient so that extra iron gets washed out from the body. So mutation within alpha and beta globin genes in India, alpha genes mainly large deletions common, minus alpha 3.7 kb, uncommon, minus alpha 4.2 kb. Point mutations are there. In beta gene, mainly point mutations, substitutions, those are insertion deletions, numbering more than 50 large deletions. This slide shows about hemoglobin indices in thalassemia carrier detections. So we can see like it's a types of thalassemia. Normal subject, beta thalassemia major, beta thalassemia intermediate, beta thalassemia minor, MCV, MCH, and HB level. Here you can see in beta thalassemia, MCV is 50 to 70 femtoliter, and MCH is 12 to 20, and normal range is 27 to 31. And hemoglobin is less than 7. Clinically popular features of thalassemia, moderate to severe, pallor anemia, mild jaundice, splenomegaly, hepatomegaly, stunted growth, typical facial changes, frontal bossing, prominent maxilla and zygoma, depressed nasal bridge, poorly developed or undeveloped secondary sexual character, typically radiological finding. This slide shows about microcytic, microcytosis, hypochromic anisopoikilocytosis, teardrop cells, target cells, which can be very often seen in RBCs of thalassemia major patients. And this is a hotspot area for mutation of beta globin gene, where you can see like chromosome IVS 1,5, frame shift 41, 42, frame shift 89, IVS 11. These are the common Indian mutations, and which are why it has been mentioned beta plus and beta 0. Beta plus is less severe. There is a little protection of at least beta-globin chain. Beta 0, when there is zero protection of beta-globin chain. So those patients are dependent on blood transfusion and chelation therapy. So here I'm going to mention about importance of XMN1 G gamma gene. XMN1 G gamma site is 5' to G gamma gene and has been associated with increased expression of G gamma, which is expected to reduce the severity of thalassemia. XMN polymorphism is a known factor and which increases fetal hemoglobin production. Among the inherited disorder of the blood, thalassemia, sickle cell diseases contribute to a major bulk of genetic diseases in India. Beta-globin mutation with the XMN1 site might be associated with elevated HbF expression, which may in turn ameliorate the severity of beta thalassemia phenotype. Here, if we are just talking in the layman language, like we can say if XMN1 G gamma polymorphism is present in a beta globin major patient, so it ameliorates the disease. Ameliorates in the sense the number of the transfusion will be less for thalassemia major patients. Gene polymorphism in children with beta thalassemia. The presence in population of 2 or more relatively common forms of a gene or a chromosome is called polymorphism. And polymorphism of G gamma globin gene is prominent. Quantitative trait loci in beta thalassemia intermedia. Intermedia is a category of thalassemia, beta thalassemia patient where the onset of the disease is quite late. In beta thalassemia major, the onset of the disease is almost the span of 6 months to 1 year of a newborn baby. Why? The reason being, when there is a switchover of fetal hemoglobin to adult hemoglobin, that gives, and when there is a mutation in adult hemoglobin, either alpha or beta, that gives to beta thalassemia major. This slide shows an association of XMN1 polymorphism with HbF induction, beta thalassemia mutations, which leads to excess alpha globin ineffective erythropoiesis, which leads to death of cell with most globin chain imbalance, which leads to anemia, increased entry into F cell program, increased erythropoietin, erythron expansion, and which is subsequently leading to the increased HbF production. That will be helpful in the amelioration of thalassemia major patients. Genetic mutation observed in beta thalassemia. Sorry. Features: thalassemia trait, thalassemia intermedia, and thalassemia major. Genetic mutation: one mutated beta globin gene is seen. In thalassemia intermedia, 2 mutated beta globin gene, one of which is a mild mutation. Sorry. For thalassemia major, 2 mutated beta globin genes are seen, and both of which are severe mutation. These are the results of our PCR RFLP, which is giving the XMN polymorphism chain reactions. And this slide shows distribution of common mutations in beta thalassemia major and intermedia patients. So in our study, we found common mutations, total a number is 79, homozygous 40, heterozygous 13. And in beta thalassemia intermedia, the number was 22, common mutation 9, unidentified mutation 13. This graph shows the common mutation in beta thalassemia major and intermedia patients. Distribution of genotype frequency of XMN1 G gamma, gene polymorphism in beta thalassemia major intermedia. This is the allelic distribution, homozygous CC. We found like 54%, 68.35%. In beta thalassemia, it was 15. Heterozygous CT, 21%. In beta thalassemia intermedia, 7%. 31.82, that is among 22, only 7. Homozygous mutation TT, 4. And allelic distribution CNT is 129, beta thalassemia major, that is 81.6%. In intermedia, it is 84.1. Allelic T, 29, 18.35, and 15.91. This is a graphical representation of previous slide, which shows a homozygous CC, heterozygous CT, heterozygous TT mutations. Allelic response in beta thalassemia major and intermedia. So after completion of study on XMN factor, we focused on haplotype of different kinds. We found haplotype of different patients coming to the pediatric department of our medical college. And what are haplotype? Haplotypes are the collection of alleles from neighboring loci and having biological significance. It usually refers to a set of significant nucleotide polymorphism within a single gene. The haplotype of beta thalassemia gene is helpful in determining genetic variation in a particular inhabitant, dependent on number of haplotype. In beta-globin gene, which consists of 5 expressed genes, that is epsilon, A gamma, G gamma, delta, and beta, and 1 is pseudogene. This figure represents 7 RFLP sites, which these are the RFLP marker sites determined by loss or gain of recognition sites onto restriction enzyme. The PCR product digested by restriction endonuclease enzyme constitute a positive plus, and the absence of a cut fragment was shown as a negative. The total number of 125 thalassemia major children were included for the study, among which 5 common mutations, that is quite prevalent in northern part of India, that is IVS15G2C, was found in 73 patients, 619 base pair deletion, that is in 28 patients, IVS15G2T, found in 17 patients, codon 4142 in 5 patients, codon 89 in 2 patients. So this shows about the association of 5 common mutations linked to different haplotypes. In our study, this chart is quite interesting because we found around 15 different haplotypes in 125 patients. And this is the overall frequency. And the HING2, HING3, HIMF1, AVA2, and BAMH1 are the restriction enzyme used for the PCR RFLP to study haplotype analysis. So here we are, still the study is going on. We are trying to correlate with which haplotype our population belongs to. So this study is still going on. We are working on it. And definitely in future, we are going to publish that. And summary of our study is beta-globin mutation with XMN1 site might be associated phenotype. As I discussed, in our study, we have found that a common mutation could be identified in 61.3% of children with beta thalassemia, and IVS15G2C was the most prevalent mutation among the homozygous mutant TT genotype. The average transfusion frequency was significantly lower as compared to CT-CC genotype. This slide shows about the psychological status of thalassemia patient and their family. Majority of thalassemia patients suffer from depressive symptoms and mental disorder as well. At least 50% of transfusion and non-transfusion dependent thalassemia patient demonstrate some impairment in quality of life. That is there. Children, teenager, families that have patient with thalassemia are more susceptible to facing emotional and behavioral problems, including anxiety and depression. Sorry about that. Thalassemia patients have significantly lower attention, verbal memory, capability, and executive functions. In addition, some new studies shows possible relationship between thalassemia and many neuropsychiatric problems such as dementia and autism, because they go through, even I'm seeing these patients from past 17 years, and I'm seeing not only the patient, even their family members, sometimes they fall into depressions. And I keep on being a genetic counselor. I do give them counseling and advisors as well. Initiative taken by Government of India for betterment of thalassemia patients' life, the Right of Person with Disabilities Act 2016 has been recognized person with blood disorders, so that thalassemia, hemophilia, and sickle cell disease as person with disability. The National Health Mission, Uttar Pradesh, India, has started free treatment as well as transfusion and management of thalassemia patients in 7 state medical colleges. And our medical college is one of them, and we are quite fortunate. At present, 2 thalassemia. Every year we are organizing the symposium come webinars for the patients that will be beneficial for them. There is a direct interaction of patient with the clinicians and counselor, genetic counselor, pediatricians, so that they'll get benefit out of that. These are the initiative taken by our lab for the eradication of thalassemia. And this big chapter we have published in Web of Science last year. That is an early diagnosis of thalassemia born to a healthy society. Trust me, it matters a lot. Reason being, if this steps can be taken by any of government, so there won't be a chances to get even a diseased child. It's just negligible. So it's kind of burden to the family as well as to the country, And that's it. And thank you so much for your patience. At the end of the day, all you need is hope and strength, and hope that it will get better and strength to hold on until it does. Thank you so much. Have a great day.